Okay, yeah, thanks for that. I just want to thank the conference chair, Jill, and the other members of the steering committee for organizing this wonderful conference and giving me the opportunity to share with you all our latest research. So today I'm going to talk to you about our efforts to probe a stage of germ cell development that, for practical and technical reasons, has been largely recalcitrant to investigations, but is crucial to understanding the impact that environmental exposures have on future generations. So yeah, germ cells constitute a metaphorical bridge between generations, giving rise to all the cells, tissues, and organs of the animal. Therefore, a germ cell exposure to toxicants have the potential to pose a significant risk to organismal health and well-being if they perturb normal germ cell function, and the memory of this can be transmitted. Now, Mike and the other speakers preceding me will give very nice introductions on the concept of multi-generational exposure, so I won't take too long on this. This illustration is taken from one of Mike's review articles illustrating the situation in females, whereby a transient environmental exposure of a pregnant female at F0 actually impacts two generations, the F1 animal as an adult and the F2 by virtue of any impacts on the germ cells within the F1. If the consequences of this are felt in the F3 generation, then the effect has gone transgenerational. So it follows then that it is vital that we understand the effect of environmental exposures on the earliest stages of germ cell development. Much of our understanding is derived from a large body of data accumulated from mouse studies over the past 4 to 5 decades. In the mouse, primordial germ cells, PGC precursors, are specified at embryonic day 6.75. Between this point and about embryonic 10.5, PGCs migrate and proliferate from the mid-hindgut through the mesentery into the genital ridge, the site of the future gonad. During the specification and migration phase, key inductive signals, notably BMP4, and the activation of germ cell transcription factors such as BLIMP1 and STELLA are crucial for normal PGC formation and execution of the correct developmental program. This program involves expression of not only transcription factors, but large-scale changes in chromatin that impact the epigenetic content in these cells, so-called cellular reprogramming. This illustration from a review article by Mitsunori Saito and colleagues summarizes the epigenetic changes that occur in PGCs. Two major reprogramming events affect DNA methylation and post-translational modification of histones. In the case of DNA methylation, PGCs undergo a gradual global reduction in CG methylation. For histones, there are characteristic changes that occur. For example, there is a concomitant reduction in a mark associated with repressive chromatin, H3 lysine 9 mono and dimethylation, as well as an increase in the mark associated with active chromatin, H3 lysine 4 dimethylation. Now, as I hope this introduction has made apparent, working with PGCs has traditionally presented technical and practical challenges. As well as being difficult to isolate, they only exist for a short period of time during a defined window of development. Their numbers are limited. It's estimated that in embryonic day 13.5 embryos, there's only approximately 25,000 PGCs present. From a toxicology perspective, a key requirement is the ability to subject cells to toxicants of interest at known concentrations and for defined durations. This is practically impossible to do with embryos within pregnant dams. A further technical challenge is that in animal-based studies, toxicants of interest are likely to affect multiple tissues and organs, which further obfuscates whether any effects that are observed are due to direct or secondary effects on the cell types of interest. Now, a major development occurred about a decade ago now with the work by groups such as Mitsunori Saito and others, defining culture methods and formulations enabling the generation of PGCs in vitro. And it is his group's system that we've adopted in the lab. Essentially, it involves a two-step differentiation process whereby embryonic stem cells are induced into epiblast-like cells, or EpiLCs, for 48 hours before forming aggregates or organoids and allowing them to grow in tissue culture for up to 5 days. In addition, the cells used carry fluorescent reporters controlled by the promoters of two germ cell markers, BLIMP1 and STELLA. These are termed BVSC cells. This micrograph here is taken from the original paper describing this system, and you can see cells activating the reporters over the course of 6 days within the aggregates. The germ cells formed using this method, termed primordial germ cell-like cells, PGCLCs, have been characterized both in terms of their transcriptional and epigenetic modifications. When extracted from day 6 aggregates, they recapitulate PGCs extracted from E9.5 embryos indicating that PGCLCs do indeed undergo correct cellular reprogramming. Now, to test the sensitivity of early germ cells, we have been working with an endocrine disruptor that obviously most of you are familiar with, Bisphenol A. It's widely used as a plasticizer, and despite being substituted out with alternatives, can still be found in many everyday items that humans are exposed to, including the lining of canned food products, dental sealants, and thermal receipts. A large body of data indicates that at moderate to high doses, BPA is a reproductive toxicant across numerous species, and in mammals in particular has detrimental effects on gametogenesis. Despite all of this, and for the reasons outlined earlier, it remains unclear what the effects are on early stage germ cells, particularly at low physiologically relevant doses of BPA. So we hypothesize that early stages in germ cell development are sensitive to environmental exposures, which will be apparent in changes in their normal behavior. Because of their unique function, serving as a bridge between generations, we propose that exposure will result in a memory with consequences evident at later stages of germ cell development. We set out to test this using BPA exposure and the PGCLC system and to identify the mechanisms involved in any perturbations uncovered. This illustrates the exposure strategy initially adopted. ES cells were cultured for 3 days, followed by harvesting and changing into EpiLC media. Cells were allowed to recover overnight before being exposed to BPA. Now, something to quickly point out is that rather than dissolving BPA in a traditional carrier such as DMSO, we actually work with BPA dissolved directly in autoclaved milk water. The 1 to 100 nanomolar concentration range is thought to encompass the physiological range of BPA detected in serum, urine, and reproductive fluids in humans. After 24 hours of exposure, EpiLCs were harvested and either subjected to standard counting using an automated hemocytometer to enumerate cell numbers, or fixed, permeabilized, and stained to examine cell cycle, proliferation, apoptosis, and DNA damage by flow cytometry. So firstly, exposure of EpiLCs for 24 hours resulted in a significant dose-dependent increase in the number of cells recovered, suggesting increased proliferation, reduced apoptosis, or a combination of both. So using the staining approach described, we were able to examine the cell cycle. Here you can see representative FACS contour plots stained with DAPI and labeled with anti-BRDU antibody following a 30-minute pulse with BRDU before cell harvesting. BPA exposure results in a significant dose-dependent increase in the proportion of cycling cells, consistent with the increase in cell numbers we observed. In terms of gamma H2AX staining, the interesting observation was that the proportion of all cells positive for this was significantly increased in BPA-exposed cells. This is seen in both cells that are proliferating, but also in cells that are viable or not undergoing apoptosis, as determined by C-PARP staining. A further interesting finding was that BPA exposure seemed to cause a slight but significant reduction in the proportion of cells undergoing apoptosis. So we next examine whether EpiLC exposure had any consequences on PGCLC formation. This slide again shows the dosing strategy used, but instead of analyzing EpiLCs, aggregates were set up and cultured for 5 days before collecting, dissociating, and analyzing by FACS. And just as a reminder, the cells used are BVSCs, which carry these fluorescent reporters to allow us to identify PGCLCs. These are representative pseudo-colored dot plots of cells from aggregates looking at the expression of the reporters mentioned. And within the aggregate, the majority of cells do not express either reporter. Developing PGCLCs initially activate the BLIMP1 reporter, followed by the stellar reporter. So there's a transition from double negative to BLIMP1 single positive to BLIMP1 stellar double positive cells. Now, these plots illustrate that whilst overall proportions of the different cell populations don't appear to change, the absolute numbers of cells, notably double positives or PGCLCs, actually do increase in day 5 aggregates. So this indicates that despite only being exposed to BPA for 24 hours, 5 days later, when growing as aggregates, this results in an increase in the number of PGCLCs. To examine in more detail the molecular changes caused by BPA exposure on PGCLCs, we performed RNA-seq on PGCLCs purified from day 5 aggregates that were either untreated or exposed to 100 nanomolar BPA. Now, analysis of PGC marker expression revealed that exposed PGCLCs do not have significant differences compared to untreated cells. However, principal component analysis indicates a clear divergence in global gene expression. And when we perform differential gene expression analysis, this reveals that while 50 genes are downregulated, almost 300 genes are upregulated in exposed cells. So when we perform gene ontology analysis to investigate biological functions and pathways affected in exposed germ cells, we observed that upregulated genes were enriched in biological processes such as coagulation, lipid transport, lipoprotein metabolism, and steroid metabolism. This latter finding is interesting in light of previous results from the Allard lab examining the effects of BPA exposure in C. elegans. In this instance, BPA was found to interfere with cholesterol transport, suggesting that there might be an imbalance in mammalian PGCLCs as well. In addition, this finding underscores the high conservation of the pathways of processes impacted by BPA between nematodes and mammals. And PGCLCs represent the immediate precursor stage before sexual differentiation. In the case of male gametogenesis, cells enter mitotic arrest and differentiate into prospermatogonia or gonocytes, while in females, cells enter meiosis I. The transcriptional alterations in PGCLCs caused by prior BPA exposure prompted us to examine whether there was any precocious entry into meiosis. The BVSC cells used in the studies presented are genetically female, and it's known that retinoic acid signaling is necessary to induce the expression of premeiotic and meiosis-associated factors such as STRA8 and REC8. Existing literature suggests that the biological consequences of BPA are mainly mediated through the estrogen hormone axis. That said, there is some evidence that in an in vitro system of differentiation involving mouse embryo body formation, BPA reportedly enhances STRA8 expression. We therefore examine whether there might be any changes in genes associated with retinoic acid biosynthesis and/or signaling and/or precocious activation of meiosis-specific programs. So when we looked, we only found that there were increases in expression of genes in 2 gene ontology terms associated with retinoic acid biosynthesis, as well as an increase in genes associated with retinoic acid response elements, and therefore inferred to be targets of retinoic acid signaling. However, when we looked at meiosis-associated gene sets, the majority did not show significant differences, and those that did actually showed reduced expression. Indeed, when we looked specifically at STRA8 expression, there's actually a trend towards reduced expression. But despite the potential increase in retinoic acid signaling, I think it's important to stress that BPA is obviously not retinoic acid, and therefore it's unsurprising that this is uncoupled from activation of meiotic pathways and processes. We also examined expression levels of all annotated nuclear hormone receptors, and several were found to be upregulated. One that I want to draw your attention to is HNF4A, which was identified through differential gene expression analysis. This is a box scatter plot of qRT-PCR data recapitulating the RNA-seq data. We also believe that it may be a general target upregulated by BPA. Again, data from a previous collaborative study involving the Allard group found that this nuclear hormone receptor is upregulated in adipose tissue, hypothalamus, and liver of BPA-exposed mice. So some of you are probably wondering about the genes found to be downregulated. Well, gene ontology analysis revealed enrichment of genes associated with gametogenesis. Several genes are members of the X-linked lymphocyte-regulated XLR family, which generate coding and non-coding transcripts that are highly expressed in meiotic germ cells. This violin plot, for example, shows XLR4. A further curious thing we observed was that a substantial proportion, 30% of downregulated genes, were X-linked. This chromosome ideogram shows the location of all the downregulated loci identified through differential gene expression analysis. Now, comparison of autosome normalized median expression values of all genes separated out by chromosome revealed that BPA-exposed germ cells had a significant reduction for X-linked genes. So together, these results indicate that BPA exposure seems to preferentially impact the expression of X-linked loci. Finally, a key requirement during PGC development is the silencing of repetitive elements found throughout the genome. Analysis of read counts mapping to all annotated mouse repeats listed on repeat marker revealed that 180 repeat elements were upregulated in exposed germ cells. The majority of these fell into these 5 families, which are evolutionarily young and therefore considered active elements. Notwithstanding the harm that retrotransposons can cause through their insertion into coding sequences and therefore impact on normal development and organismal well-being, their silencing is also required for normal germ cell formation. So to summarize the main points I presented, using this in vitro model system, we've shown that EpiLC exposure to BPA causes dose-dependent increase in cell number, associated with an increased proportion of cells in S phase, as well as a reduced proportion undergoing apoptosis. Exposure also results in a greater proportion of gamma H2AX positive EpiLCs, consistent with increased DNA damage, and obviously a potential source of genetic mutation. Exposure during the EpiLC stage results in increased proliferation of PGCLCs, indicating the existence of a memory of this exposure. PGCLCs derived from BPA-exposed EpiLCs have an altered transcriptome, with significant decrease in expression of X-linked genes and increased expression of retrotransposons. Differential gene expression analysis reveals deregulation of genes involved in lipid metabolism and cholesterol homeostasis. And therefore, prior exposure has likely to have significant impacts on early developing germ cells. So I just want to acknowledge the members of the lab that contributed directly to the study, specifically Hui Jiang and Yanni Kang, and obviously Patrick, and all the other members of the Allard group. And these are the funding agencies that made this work possible. Thanks for your attention.
Great, thanks. That was really interesting, Steen. I think these PGCLCs are really going to be incredibly useful moving forward to figure out how all of these exposures influence germ cells. Any plan to maybe apply these findings back to the in vivo setting and looking at the developing germ cells or primordial germ cells in response to?
Well, so we're not really an animal lab where we do experiments in animals. We're purely using the in vitro system. And one of the strengths of it is that by using these female cells, Mizunori Saito has published an extended protocol that can allow you to push it out into growing oocytes. And we would be very interested, to see whether the exposure at the PGCLC stage does then have these impacts if we push them out to oocytes,
Yeah, I think it's a great, especially like Jill mentioned earlier, with the oocytes where they're so hard to get. I mean, you can get maybe 30 from a mouse. If you can grow them to in the hundreds and even tens or hundreds of thousands, it would be an incredibly useful sort of substrate to do these experiments on, to study this from the female germline perspective. Mm-hmm. Great, thank you so much. I think we are going to take a break, and then we'll follow up with our second part of the session on DES.